Hi everyone. Thank you for joining us for this Q&A AMSs for Health Data Science seminars. This is organized by the Startup AI Alliance. So here's just a brief introduction. So the group aims to organize activities that empower physicians to participate more actively in AI research and leadership. So here is the website, and then right now it's recognized as an ASA interest group. And then we also upload the recordings of the seminars to this YouTube channel. Here is the list of the steering committee, and then currently we just have the first of your officers. So for today, it is my great honor to introduce our speaker, Dr. Russell Poldrack. So actually, Dr. Poldrack is the first speaker we invited from outside statistics. Thank you so much for joining us, and we are really excited to hear from you about your research. Dr. Poldrack is the Albert Raylong Professor of Psychology at Stanford University and serves as the department chair since 2023. He also holds a position in the Department of Psychiatry and Behavioral Sciences. He is the director of the Center for Open and Reproducible Science and the associate director of the Center for Data Science. Dr. Poldrack's research interests include using neuroimaging to understand the brain system underlying decision-making and executive function. Dr. Poldrack is a leader in advocating reproducible neuroscience. His group has developed neuroinformatics tools to help improve the reproducibility and transparency of neuroscience, including influential open tools and infrastructures like Cognitive Atlas, Open Neuron, Neuron Vault, and then standards like BEAST, the brain imaging data structure. Actually, the first fMRI data set I work with was downloaded from Open Neuron, which is called OpenFMR at that time. So it is a very good starting point for beginners who wanted to step into the field for neuroimaging. So it is very easy to access and download the data, and then it has multiple data designs. Dr. Poldrack has received numerous honors and awards. Here I only listed a few, so I won't name those. Now, without further ado, I will hand it over to Dr. Poldrack. So before that, just some logistics for today. So please save your question to the end of the session. So we should have some time for discussion. And then, yeah, Dr. Poldrack, please take it over.
All right. Thank you very much. Thanks for the introduction and the invitation to be here. So yeah, I'm going to talk today about work that we've been doing over about the last 15 years, trying to build credibility in neuroimaging through open science practices and tools. And the first question you might ask is neuroimaging research credible, right? I mean, you see every day new publications, both asking basic science questions and also sort of clinical questions. This is just a plot of the number of papers that mention, each of these different diagnoses. And clearly there are thousands of papers using fMRI to examine each of those. And, every week it feels like you see, a new headline about, some way of using brain imaging to try to understand how we might treat these disorders. But there's also been evidence that, a lot of the research that's been done doesn't really add up. This is a kind of a well-known meta-analysis from a few years ago that basically reanalyzed previous meta-analyses, took 99 studies, all of which individually claimed to find differences between depressed and non-depressed individuals, put them all together and found no convergence. So clearly there's a problem there. You may also be familiar with sort of statistical questions about, how fMRI data are analyzed. This is a very well-known paper from back in 2016 by Anders Eklund and Tom Nichols, which basically showed that many of the tools that were being used to do correction for multiple comparisons in fMRI were not actually performing as people thought they were. So, there's been a lot of concern about the credibility of the research that we've done in my lab for a long time, and of that kind. And so we've been trying for a long time now to kind of improve that, and with the intuition that transparency and a focus on reproducibility are really the ways to do that. And we refer to that as an open science revolution. And so I'm going to talk about kind of three different things. I'm going to talk about open data, the platforms that we've been involved in developing for sharing of data. I'm going to talk about open source software, and then I'll talk about the standards that underlie particularly open neuro and open source software, particularly this thing that was already mentioned called the brain imaging data structure, or BIDS. So, one thing to say is that in some ways, neuroimaging is sort of a best case scenario for open science. You know, I'm going to be focusing today primarily on magnetic resonance imaging. And this is a tool that's widely used by neuroscientists to study human brain structure and function. And the nice thing about the data is they're digital end to end. We get them off the scanner digitally. There's occasionally, you know, manual annotations, but almost always all the data are digital. Most of the analyses are automated or automatable, at least. There's usually very few or zero manual analysis steps. And, you know, for more than 25 years or for around 25 years, we've had a standardized image format. So all the software packages in our field use, at least can use this nifty image format. And we also have a common spatial coordinate system that basically lets you describe in 3 dimensions where you are in the brain. And I'll say more about that in a moment. Now, interestingly, we had a false start in our field for data sharing back in around the year 2000. A group started this project at Dartmouth called the fMRI Data Center, which was meant to basically share raw fMRI data. And because the founder of this, Mike Gazzaniga, was a kind of a very big figure in the field, he got some journals, particularly Journal of Cognitive Neuroscience and the PNAS, to require researchers to upload their data to this database in order to get published there. And you know, there was a firestorm basically. There were, you know, editorials written in Nature Neuroscience and in Nature. Then the text on the right is from an open letter that was sent around by some very, you know, big names in the fMRI field, you know, highlighting that they're particularly concerned with any journal's decision to require all authors of all fMRI related papers to submit all their experimental data to the data center. So the field just wasn't, we had the tooling, but we didn't have the kind of, you know, the social engineering that we needed. You know, in my view, 2010 was kind of the year when data sharing really became a thing in brain imaging. And this paper was certainly, you know, one of the big reasons for that, by Bharat Biswal and Mike Millam, which basically brought together 35 centers around the world and amassed over 1,400 individuals worth of this particular type of resting functional MRI data, and show that you can make discoveries using this big data set that you could never make using small data sets. And that really kind of got the ball rolling. Now, I think it's fair to say that data sharing, I don't know if we'd say it's become the norm. It's certainly becoming the norm. We wrote about this a few years ago in Annual Reviews of Biomedical Data Science. Mike Millam and colleagues wrote about it in 2018 in their Nature Comms paper. But, you know, both highlighted that there's an increasing amount of data that's available of different types, and I'll talk about these different types. And those data are being increasingly reused. That's what's shown on the right, is the number of publications using these different data sets that Millam's group has shared. It's funny. Back in 2019, so Organization for Human Brain Mapping is the main meeting that we go to for kind of fMRI methods. And one of my colleagues overheard a very senior researcher saying that OHBM has been taken over by the open science zealots. And interestingly, that senior researcher has now become a total open science zealot. So I think it is fair to say that, you know, a lot of people have been sucked into this ecosystem. So I'm going to first talk about what you might think about as the ecosystem for data. And I'll talk about 3 different ways of sharing data, of 3 different types of data that can be shared, that span this kind of spectrum from high breadth and low depth to low breadth and high depth. So I'll talk about each of these projects in turn. So on the one side, the Neurosynth project focuses on sharing what we call activation coordinates. So when people publish papers in our literature, they very often put in tables like the one you see here with XYZ coordinates. And these describe a location in the brain using this sort of kind of standardized coordinate system that tells you where is that activation. So if somebody says there's activity at -52, -54.9, I know where that is in the brain. And lots of papers have this. Tal Yarkoni came up with a way to basically automatically extract those from publications and develop this tool. We worked with him on this called Neurosynth. You can find it at neurosynth.org. And basically, it's a database that uses those published coordinates to create meta-analytic maps. So if you want to, in particular, mapping terms from the abstracts into brain activation. So if you want to know what are the brain areas that are associated with the term working memory, you can put that term into Neurosynth and get back a map that you can download or, you know, kind of poke around on. we showed in the original paper that you could use this to do a number of interesting analyses. So for example, let's say that you want to know, which is more likely that you have some data from a study and you want to know, is it more likely that it's a study of working memory or emotion or pain? You can basically build a classifier based on the presence of those terms in the abstracts and do a pretty good job of classifying new studies. We had new studies that were labeled as being about working memory, emotion, or pain, or even new individual subjects could be labeled accurately, and you get maps that kind of look reasonable given what we know from the literature. We took these data to subsequently go ask, can we start to use natural language processing to understand sort of at a more general level, what are the concepts in this literature and how do they map to a brain system? So we took, in this case, we used the full text of the database with almost 6,000 papers and fit topic models using latent Dirichlet analysis, latent Dirichlet allocation, and then basically filtered the terms by either whether they were about mental functions or mental disorders using that cognitive atlas ontology that was mentioned earlier. And this allowed us to create what we call topic maps that show basically a set of brain systems that are associated with a set of terms that are about different things. So for example, the first topic here, these are the highest terms. These are kind of language related things, and we see it's mostly activity in the left is right here as sort of is the standard left hemisphere language areas. Here's several that are about kind of emotion, empathy, pain, awareness, and you see activity in areas associated with that. So you basically see sets of terms that are related to one another that are also related to a set of brain areas. And that's become a really useful tool moving forward for kind of getting an understanding of what particular systems in the brain do. More recently, we worked with Gail Veracqua's group, work led by Jerome Dacus, to create kind of an improved version of Neurosynth called NeuroQuery. It's both improved on the data side, but particularly on the modeling side, because it uses a more sophisticated model for mapping between terms and brain areas, particularly by doing this sort of query expansion. And what this does is it's similar to Neurosynth, but it provides actually a much better performance, particularly for terms that are relatively infrequent. So you can see here what the maps look like for NeuroQuery versus Neurosynth or versus this other kind of topic modeling related method. So that highlights the utility of those kind of very coarse data that you can just get from papers, right? And I think the current NeuroQuery database, which is a bit larger than the NeuroVault, has on the order of I think above 18,000 papers in it. So quite a bit of data in that database. Now, often you want the real data from a paper, not just a very sparse description of where the center of activation was for each of those clusters. And so we also, a while ago, led by Chris Gorgolewski, who's now at Anthropic, developed a tool called NeuroVault, which allows people to upload the whole brain statistical maps that are kind of like the main statistical result of a neuroimaging study. We do usually for many studies, we'll do some kind of linear modeling analysis at every voxel. We call this mass univariate modeling, and then we'll have a map of say 200,000 voxels, each little cube in the brain, and a statistical value at each of those, and we'll do inference over that. What we'd often like to do is actually see that raw map, not just see the figures that are made. And so what NeuroVault does is allows people to upload and share those statistical maps. Here's an example of a T-map from a particular study that we had shared. Currently, it has about 16,000 collections. Usually a collection maps to like a publication, with about 64,000 maps. And one of the nice things is that, this gives, each image has a unique persistent identifier. So if you're publishing a paper, you can put this identifier in, and then anybody who was looking at your paper doesn't just have to look at your figure. They can click on the identifier and it'll take them to the data set. They can download it, they can poke around on it and, threshold it at different values and kind of really see what the data look like in a way that you can't from a publication. Here's an example of the reuse of NeuroVault. This is work done by Bertrand Thirionn's group, led by Raphael Minuet, where they basically took NeuroVault maps and the concepts from that cognitive atlas ontology that I mentioned before, and basically did a couple things in parallel. They did a sort of like a enrichment of the concepts using natural language processing, and then they did processing of the imaging data, and particularly kind of embedding it in a low dimensional space, and then asked how well can you decode the cognitive functions that you think are going on in an imaging data set from the data themselves. And what they showed is that compared to Neurosynth and this other GCLDA method, they could decode much better data from individual subjects who are doing all these different tasks, and see what in fact are they doing, right? So for example, for some things you can decode really well. For example, are you pressing your left finger or not? And that is associated with activity in the right motor cortex. So some of those things are, are you looking at a place or a face? Some of those things can be accurate, can be decoded with very high accuracy in individual subjects based on this classifier, this kind of meta-analytic classifier, using neural networks that they trained based on those data. So I'm going to focus most of my time today talking about the sharing of raw and processed, but fully processed data sets, which is the goal of the Open Neuro project. So Open Neuro began life, as was mentioned, as a project called Open fMRI back around 2012, I think it actually went online. We transitioned to the Open Neuro name in 2017 to reflect the fact that we were generalizing out just from functional MRI data. And it's grown significantly over the number of years, up now over 1,400 data sets, over 63,000 people's worth of data, mostly humans, a few various other critters. And one of the underlying kind of intuitions and drivers of what we've done in Open Neuro is the idea that simply sharing data is not sufficient. You need to share it in a way that makes it useful. And we're inspired by what have come to be called the FAIR principles. That data should be findable. You should be able to go out and figure out where it is. It should be accessible. Once you know where it is, you should be able to know how can I get the data. It should be interoperable, meaning once you have the data, can you actually work with them? And it should be reusable, which primarily means, do you know what your rights and responsibilities are for reusing the data? And so one of the really important aspects of this is having the data structured in a format where somebody can take them and just use them without having to ask questions about what's what. So starting in 2015, we began to develop this thing called the brain imaging data structure. It's now become a full community organized project. It has a steering group and that's elected every few years. And so what it describes is really two things. One is how files are organized and named. And you see that usually when we get data off of an MRI scanner, they're named in this kind of inscrutable way on the left. This is what the names look like you have they follow a convention that can be machine readable. And then we also have a metadata standard that basically says in these JSON files that are associated with each image, what's the metadata that should be there? Or in this file that describes all of the participants in the study, what's the metadata that should be there and how should it be described and formatted? We published a paper last year that kind of laid out, where BIDS came from, where it's going, and highlighted, the growth of the standard. This is just one example from, I'll talk later about MRIQC, which is a tool we developed that uses BIDS data sets, and it tracks how many unique data sets have been run. And I don't have the newest data, but as of a couple of years ago, several 100,000 data sets have been run, and we're pretty sure that it's probably approaching 1 million data sets at this point. One of the unique aspects of BIDS is it was built to be extensible, kind of inspired by the Python extension proposal protocol. And so over time, it started with MRI, but it's now come to encompass many other types of data, including magnetoencephalography, intracranial EEG, scalp EEG, positron emission tomography, NIRS, microscopy, a bunch of different types of data. Lots of people have been involved in this organization, as you saw by the number of names on the paper. So if you go to Open Neuro and you want to upload a data set, you first log in, and then you are asked to select the folder that you want to upload. And we have a validator for BIDS that works in the browser. So what it will do is when you select a folder, before it ever uploads any data, it will look at the structure of those data and say, do they meet the BIDS standard? And if they do, then it just uploads them. If it doesn't, then it tells you what you need to fix in order to make it upload them. And then you have to add a bit more metadata, and then you can basically very quickly share your data. And the data are shared completely openly under what's called a CC0, like a public domain dedication. So they're basically you can do anything you want with them, except for trying we make you sign a click-through agreement saying you won't try to re-identify the subjects. This is the growth of that database over the years. And you can see that it's grown. The number of data sets has just grown like, really consistently over time, with a few big jumps in the number of subjects when big data sets get uploaded. But for the most part, just continued growing over time. Here's an example from our early work of reuse of open neuro data in the context of AI models. So, we think that obviously, AI models are very data hungry. And this is one of the largest data sets available for, for functional MRI data or other types of MRI data, particularly one with very liberal, permissions to reuse. So Armin Thomas in our group wanted to look at developing what we call foundation models, like pre-trained models using a self-supervised objective that we could then use, just as you can use ChatGPT to write poetry, even though it wasn't trained to do that. We could try to use these models to then do other things like decoding a brain function. So he tried a number of different architectures, and ultimately we kind of decided that this transformer-like architecture we call causal sequence modeling back then basically was kind of the best one. And had to do a bunch of work to figure out how do you embed the timing of the scanning in the sequences and so on. But then what we do is we take that pre-trained model and apply it to new data. In this case, applying it to data from the Human Connectome project. No, sorry, this is actually from a different project, this multi-domain task battery. And basically showing that you can do much better at decoding what a particular person is doing at a particular point in time from data using the representations learned by this foundation model compared to the kind of more standard linear representations that had been used for a lot of decoding work, prior to this. And now there's a whole cottage industry about building foundation models for functional MRI, and a number of them are using data from Open Neuro. This was using a relatively small amount of data, but we're working, as I'll talk later, we're working on generating a lot more. So that's sort of the data ecosystem. I want to now turn to the software ecosystem. And first, so this has come to be called the NIPREPS ecosystem. It's a bunch of tools that kind of work similarly. They're almost all kind of BIDS centric. They all work with BIDS data. But I do want to highlight the fact that we're building on top of open source projects that have been going on in our community since 2000, or even SPM's been around well before the year 2000. So for a long time, we've had open source projects in our field, and we're kind of building on top of those. So the main tool that we have developed is this thing called fMRI Prep. And so the figure here highlights the fact that when you get data from a scanner, that's the stuff out here on the top, a ton of pre-processing has to be done to make those data ready for statistical analysis. And depending on the data, there are different things you might want to do. And so what we've tried, and the way that this has worked in the past before fMRI Prep is every lab had its own kind of pre-processing workflow. Most of them were based on a single software package, and most of them relied on a shell script that had been written by a postdoc at some point that everybody's afraid to change because they know it'll break and they don't know how to fix it, don't quite understand how it works. And so we wanted to build something much more robust, and also something that could basically adapt to the data set and do as the best possible job of processing a data set using the best tools possible, even if it requires pulling from different packages. We also wanted to put the data out in a way that was standards compliant. And so one of the things that was developed in the BIDS community was called the BIDS derivatives format, which is basically a scheme for describing and organizing the outputs of a statistical analysis. Like before this, most software packages just decided willy-nilly how they wanted to organize the output, and you kind of had to figure it out. And often the names are not, kind of human readable at all. So BIS derivatives tries to move towards a machine readable and human readable format for the organization of those outputs. We also, the problem with a powerful tool like this is it can become a black box for people who don't know what they're doing. So what we try to do is make it more of a glass box by providing visualizations at the points where we know things are likely to go wrong, and provide a lot of guidance to how to identify problems at those steps. Now, when we build software, we often put telemetry in it to basically know how many people are using it, how successful is it. And so we've had that, for several years now. And this shows the usage, the weekly usage of fMRI prep. And so you can see that, for the last 3 years, basically, we've had, sustained usage of about, over 8,000 successful runs per week. And a really consistent, at least over the last year or so, average weekly success rate of about 65%. And we think that most of the failures are likely to be problems with the data that's being put in, not necessarily problems with the software. But the nice thing about the telemetry is if it crashes, we know why. And we can, if we put out a new version and we suddenly see a bunch of crashes, we can go look at what's going on. So it really helps us create kind of more robust software. We've also built another tool called MRIQC. We call these BIDS apps because they're applications that are kind of natively support inputting BIDS data. MRIQC addresses this problem that many people have had for a long time of how can we do good quality control on MRI data? And Oscar Esteban, who's now in Switzerland, developed this tool. So one of the things that we've been doing recently is basically figuring that anybody who wants to use preprocessed data shouldn't have to download the raw data from OpenNeuro and then go process them themselves. That's a big waste of energy, a big waste of time. And so we're processing all the relevant OpenNeuro data sets with both MRIQC and fMRI prep, and then releasing those openly on, this is a screenshot from the OpenNeuro data set showing how you can pull them either using the AWS tool or DataLab. We currently have 306 processed data sets with both fMRI prep and MRIQC, 10,000 subjects and over 15 million time points. And I want to highlight, for those of you who build software, this is also a really good stress test for your tools, like running a lot of data through it. And this is just one example. So we've been doing a lot of this processing at the Texas Advanced Computing Center, where we have an allocation on their Frontera supercomputer. And we basically got shut down. And this is the email telling us, sorry, we had to revoke your access. We're seeing activity up to 40 million files in less than a 24-hour span, which is putting an unreasonable strain on the file system servers. This at the time was the biggest academic high-performance computing system in the country. And so we basically were breaking that biggest HPC system. And so what that drove us to do is actually change our model for how we do the processing, to move away from this monolithic thing where we basically generate all the outputs anyone might need, to basically what we call a fit transform model inspired by scikit-learn, where we first generate the minimal set of derivatives that you would need to deterministically regenerate any of the other outputs that someone might want. And this greatly reduces the number of files. You can see that the size of the work directory went down by 38 gigabytes. And the total amount went down by 41 gigabytes, and the number of files goes way down by 78%. And so we've really helped kind of streamline processing and also reduce the amount of resources, the power resources that go into it. So you might be asking, you've talked a lot about pre-processing, what about statistical models? And so this is something that we've been working on a lot in the last year. So as I said earlier, most kind of task fMRI studies, like the ones that we do in my lab, run these mass univariate linear models. So basically, usually just a standard GLM or maybe a fancier model at each voxel, over 200,000 of those, and then do some kind of inference over that. It turns out there's a lot of variability in how people do this across different research groups, and it's often actually difficult to tell what somebody did just from the paper. So I'm going to show an example from a study that we published back in 2020, led by Tom Schoenberg in Israel, and with our statistician colleagues, Tom Nichols and Jeanette Mumford, where we basically gave out a data set to a bunch of different groups. We collected this data set. I'm not going to go into the details of how we collected it. Tom collected this data set with 108 subjects at Tel Aviv University. We gave that data set out to research teams who were recruited from the kind of the domain of decision-making researchers that who would know about this kind of task. Each group could have up to 3 people. They had to sign an NDA saying basically that they wouldn't talk about their results until we lifted the embargo. They were all offered co-authorship on the initial paper, which is why the paper has about 200 authors. We gave out the raw data set to 82 teams. We ended up getting data back from 70. We gave them about 3 months to give us. The main thing we got was yes, no answers to 9 hypothesis tests, which were all of the form. Is there significant activation in some brain area for some feature of the experimental design or some contrast on the linear model? And we didn't really restrict it. We only said you had to do a whole brain analysis. We also asked them to provide a detailed written description of their analysis workflow based on this kind of scheme that we put together with Tom Nichols' leadership a number of years ago it's called the Kobadas report for describing, basically standards for describing an fMRI data a set or data analysis. Strikingly, none of the 70 teams used the same workflow. And even more strikingly, even with a detailed written description, we often couldn't tell what they did. For a number of subjects, there were errors that were made. We had to get the original outputs, the files from them that were output by their software in order to tell how they had actually built their model. This is the primary result. The y-axis is the proportion of teams saying, yes, there is a significant effect for the 9 hypotheses sorted by that proportion. So if everybody agreed, these numbers would all be 0 or 1. Either everybody would say no or everybody would say yes. You can see there's 3 where people mostly agree on no, 1 where people mostly agree on yes, and then 5 hypotheses where 20 to 40% of teams are saying yes. That's a really strikingly troubling amount of disagreement with the same data. Another way to look at this is across teams, there were 70 teams, 33 different patterns of outcomes. And for any hypothesis, there's at least 4 workflows that can give you a positive result. Nonetheless, I mentioned there were a number of teams that made errors in their analysis. Even if you include those in this image-based meta-analysis, we do see that there were kind of reasonable things based on our knowledge of the literature that came out in this analysis. And so this has really inspired us to think a lot more about how we can come to embrace this, what's come to be called the analytic multiverse. Because a few of those people made errors in their analyses. A lot of them were doing things where they had good justification, and there's often not a right choice for which algorithm to use or what threshold to use. And so we need to understand the effects of analytic variability, as people have been talking about in the literature, for at least a decade now. And know how robust our results are to analytic variability. And so I'm going to show an example that we published recently that looked at the effect of analytic variability on test-retest reliability of task fMRI data. So increasingly, people want to do analyses like correlations of some kind of fMRI signal with some kind of other phenotype, age, memory performance, intelligence, whatever, genetics. And obviously, in order to do that, you need reliable task fMRI measures. And a lot of work has previously shown, this is Elliott et al. Did a nice analysis of this, showing that, on average, the test-retest reliability of task fMRI data is pretty bad. It's generally below about 0.5. And so we wanted to know how this relates to analytic choices. And so Michael Demedico in our group did this with a specific, I'm not going to talk in detail about this functional MRI task. It's a task called the monetary incentive delay task. It's a task that's used heavily in the addiction literature because it's thought to measure kind of activity related to motivation to get rewards. So it involves the potential to win or lose money, and you have to make a response. He took 3 data sets, one subset of this large adolescent brain cognitive development data set, and then a couple of other data sets, all of which use this task, and basically looked at the effects of varying the pipeline that's used to process and model the data, coming up with 240 pipelines, each of which is based on things that are done in the actual literature of studies that have used this task. Like there's hundreds, if not more than 1,000 studies that have published on this task. And there's a real high degree of variance in how they've been modeled. And so this, at the top you see is the ordered ICC estimates across those different pipelines this is for one of the specific data sets. And you can see that it ranges from kind of about, a little bit above 0.4, kind of at the ceiling of what we expect of task fMRI, down to basically zero, like completely unreliable in a test retest sense. And so what you can see is that there were certain analytic choices, particularly around the statistical model that was used and the contrast that was used across the parameters from that model, that make a huge difference. I should add that this project subsequently got us to dig into these models, and it turns out that all of these models are misspecified. We have a paper that's in press right now, led by Jeanette Mumford at Imaging Neuroscience, that basically shows how that whole literature that's been around for 20 years now has basically mismodeled this task, and we show how that can bias parameter estimates. But even across that, you see that there's huge differences in reliability. Interestingly, if you look at the magnitude of the parameter estimates in like a brain area people in the addiction literature care a lot about, you can also see that across those different analytic choices, all of which are meant to basically measure the same thing, this kind of like motivation to obtain or anticipation of a reward. You can see massive differences across analytic workflows, particularly related to the contrasts that are used and the model specification, from like, huge, like one, a Cohen's d of above 1.5 down to a Cohen's d of below -1. And interestingly, this doesn't map directly with, if you look down here at the specification curve analysis for reliability, the parameter estimates don't map to the reliability estimate. So some models can give you, really decent reliability, but bad estimates. Interestingly, though, if we look across data sets, this is the rank, or this is the rank of the Cohen's d, basically across. These are two different data sets. This is basically the ranks of those numbers. You can see that the rank correlation is pretty high, 0.85, right? So if a model does a good job of getting a big parameter estimate on one data set, it generally does a good job of getting a big parameter estimate on another data set. So one of the challenges of this kind of multiverse analysis is scaling it to whole brain fMRI data. Both because we're generally doing this on high performance computing systems, and because it's just a massive amount of data. And so the one thing that we realized is if we wanted to do this at scale, we needed a machine readable way to write down a statistical model so that our tools could read it in and just automatically implement it without us having to kind of fiddle. And we also realized that this would benefit reproducibility, because somebody who uses a different software package than I do can give me that machine readable description, and I'll know exactly what they needed to do. So we've been for a number of years working on this project called BIDS Stats Models, which is meant to be a machine readable model specification for fMRI analysis. You can read about it here. We have a paper that's in prep right now, so hopefully we'll be submitting something. We'll have a preprint out soon. The idea is, often we do this multi-stage analysis where we'll have, each subject might have 6 runs. We analyze those individually. Then let's say we have 3 subjects. We combine all their 6 runs for each of these subjects. And then we do some kind of like group analysis, like, the simplest would just be a 1 sample t-test versus 0 at each voxel. And so what BIDS Stats Models does is provides a scheme for describing a particular model in a JSON file, basically, such that one could then take this and implement it in any particular software package you want to. Of course, you need the tooling to do that. And so we've been developing in parallel this tool called FitLens, which is basically kind of like the benchmark implementation of BIDS Stats Model. So what it does is it takes in a BIDS data set, particularly one that's been processed by fMRI prep, and it also takes in that BIDS Stats Models specification. And then it does the linear modeling and outputs results in a BIDS derivative output format. So now we can start to imagine actually running, hundreds of general linear model processes at scale on HPC, and having the outputs organized in a way and the models organized in a way that we can actually understand what's going on there and be able to model them in the way we want to. We're currently applying this to task fMRI data sets in Open Neuro. This has turned out to be a lot harder and has really highlighted some of the limitations of the metadata that we get with Open Neuro data. We've had to contact data set owners to get a lot of details. So far, we've made it through 57 data sets, almost 2,000 subjects worth of data, almost 500 contrasts. And we'll be sharing these outputs as well on Open Neuro. Now, one of the challenges with this kind of hand-built multiverse, right, is that there's this combinatorial explosion of pipeline configurations, right? If you want to do all combinations of all of these different possibilities at each step, it's just a massive amount of number of things one would have to run. And so Rasko Sirik in my group a couple of years ago adapted this idea that's been sort of emerging in the literature recently about differentiable analysis workflows, where instead of like saying we're going to have each step in the model be some particular version of that analysis, and then we try a bunch of versions of that. Instead, you treat this as an optimization problem where you have your data, you have some predictive objective, right? Like it might be task ID or age or something like that. And then you basically build differentiable versions of differentiable modules. You can think of these as like layers in a deep neural network, and each layer implements a particular aspect of the processing stream. And so the idea is you can now, instead of like trying a bunch of different things and looking how they work, if all you want to do is find the best workflow, you can just learn it from the data. Obviously, you need a lot of data. And so Rasko focused in particular on what probably turned out to be too hard of a problem. He got his PhD, so that's good. But making progress on this problem was really hard. One of the things that we do particularly in resting functional MRI data is we call parcellation, where you have to basically break up the brain into some. It's basically a dimensionality reduction process based on with some kind of spatial constraints. And he basically reframed this as an optimization problem with both spatial and temporal cost functions or losses. Combine those losses and basically use the minimization to use gradient descent to find the optimal thing. And what we find is we compare it to, for example, the reference atlas from Thomas Yeo and colleagues. We see that we get pretty similar mappings. And in fact, if we look at its performance in terms of one of the things that's often used in looking at these types of outputs is the homogeneity of the connectivity within each region. And we find that compared to this MMP, which is a kind of a previous state of the art, or a number of other tools, that our tool does better than those other tools in terms of homogeneity. It turns out that this, in general, doing this is really hard. Implementing these individual steps is really hard, and it also in sort of working on this and thinking about this. We started thinking about this. Many of you may be familiar with Rich Sutton's Bitter Lesson piece from a few years ago, which basically made the point that AI researchers have often tried to build knowledge into what they do, and that sometimes helps in the short run, but in the long run, it often plateaus or even inhibits further progress, and that in general, breakthrough progress eventually arise by basically simpler methods based on scaling computation with very large data through search, through more generic search and learning processes. And so you might ask, like, why are we doing this? With all these like hand-built little modules, when we could just do this, right? We could just take a big complex model and have it learn the relationship between those preprocessed data, or even have it learn the relationship with raw data if it can, and those output predictions. And I think this raises a really interesting question for us in the context of AI models making their way into data analysis, which is, would we trust this? And I would be a little bit worried about trusting a completely black box model. Obviously, there are ways to get kind of insight into how these models work. But I still feel, as a scientist, a lot of trepidation about trusting a black box model like this. Happy to discuss that more. So finally, I just want to talk about another aspect of open science that I think has worked really well in our particular community, which is this openness to work together. And I want to talk about a project that we started a number of years ago with several other groups, the Mike Millam's group, Ted Satterthwaite's group, Damien Fair's group, um, which basically involved trying to figure out how well we could harmonize the outputs of different pre-processing pipelines. So, there's a bunch of these. Here's just a few examples. The Mike's Millam's team has this thing called CPAC. The ABCD team had this thing called ABCD bids. Somebody else has something called CCS. We had fMRI prep. We have like a long-term support version. And when they processed the same data sets through each of those and compared the correlation matrices between different brain areas that came out across different atlases, you see like a pretty massive lack of reproducibility, right? Where the correlations, for this particular atlas, for the 1,000-piece atlas, are only around 0.5 on average, which is pretty bad. You'd like them to be very high. What Mike's group did was shown that they can, by this CPAC is like a configurable pipeline. They showed that with some work, they could actually get CPAC, when you configure it the right way, to actually behave very much like either ABCD bids or CCS or fMRI prep. They have much more reliable outputs between them. But in doing this, we also realized that, many of us, each of our groups were kind of like reinventing the wheel. We were all like building functions to do the same things, but we never shared, in part because we didn't trust that each other was doing the things in the way that we liked. So we came together and developed this thing that came to be called the InMind Consortium. InMind stands for Never Mind, This Method is Not Duplicated. And the idea was to come up with a set of standards so that we can all be comfortable sharing software components across the different labs. And this involves, one, just standards around software creation, testing, benchmarking, terminology, toolkits to actually do the testing, and then a bunch of other stuff that we haven't quite gotten to yet about, advanced engagement training, advancement, governance, that sort of stuff, because this stuff is hard. And we've, built this kind of like, starting point of like giving out badges on each of the different levels for different software packages. But this just highlights how when you're working in an open science way, you can start to reduce the amount of duplication that's happening across groups and also improve the quality of the work that's coming out. So I have the figure here because I live in a little bubble where I think everybody is doing open science. Clearly, a lot of people aren't, but I'm happy in my bubble, so I'm going to continue to live there. I do think that our field has developed this really enviable ecosystem. When I go talk to other areas, people from other areas of science, they often ask, like, how did you get there? And I'm like, well, we've been doing this for 20 something years. And obviously there's more work to do. The beauty of open science is I think there's room for everybody to contribute. And I also want to highlight the fact that the data that's coming out of these tools, I think is going to be particularly important as we think about developing, more data hungry sorts of AI models in the context of data analysis. So let me just thank everybody who has worked on this with us over the years, and I'm happy to take some questions.
Great, thank you so much. This is a really inspiring talk and then very interesting, and then also offering a completely different angle for us physicians to think about problems. I saw there's a comment from Diane Patterson. So do you want to just speak out and then unmute yourself?
Sorry, you
Oh,
are
hi.
right. Okay. Diane, do you want to say something?
Hi. Yeah,
Okay.
just been following Gary Marcus with regard to AI commentary, and he recently wrote something on Sutton and said that Sutton has kind of recanted this scaling only model and is coming over to Marcus's position that you probably need something more symbolic. That AI it's not going to get there with just scaling.
I would want to see Rich Sutton actually say that. I think that a number of people, that the bitter lesson is is a kind of an extreme point, right? But I think that even if you do decide that, we're not going to get to full artificial general intelligence via really generic models like transformers or, deep RL models, that the point still stands that in general, lots of data and a relatively general architecture has outperformed more custom architectures. Clearly that's true in language modeling, right? I mean, people spend a long time trying to build, language models based on what they knew about language, and it turns out that you don't need to know anything about language. You just need to know something about this generic transformer architecture to get you a good long way. I'm not saying it gets you to full language understanding. So I hadn't heard. I tend to tune out Gary Marcus because I think most of what he says is kind of BS. But I'll have to go find this and see what Rich actually said.
Thank you.
If you have a question, so then please raise your hand and I will call your name and then you can unmute yourself and then ask. And while we are waiting, so can I ask a question? So we see a lot of heterogeneity just due to like a modeling or just choosing the contrast in one of your examples. I'm just wondering, like as a biostatistician or statistician, so how can we help and then to try to resolve that issue?
Sorry,
So do you have
just say
any?
again. I couldn't. The sound was kind of bad on my side. Can you say again? What is the issue that you're talking about resolving?
So like we see a lot of like in terms of like a heterogeneity in terms of the output from like a modeling aspect and then choosing the contrast.
Right.
So I'm
Right,
just
okay,
wondering if you have any like insights and then recommendation for us as a biostatistician who can help. Yeah.
Right. I mean, I think the place where statisticians can help is probably in getting insight into the degree to which, I mean, there's there's at least two reasons why you might have this heterogeneity, right? One is that there are trade-offs that are legitimate trade-offs, right? And you can choose different methods depending on where you fall on that trade-off. Another is that, there's bias in the method, and some methods really are better than others if we understood that bias. And so I think statisticians can help us really think about kind of how to figure out like when is the variability really about kind of legitimate trade-offs versus when is it about biases. And I think like the work that I mentioned that with Jeanette Mumford, who's a biostatistician in my group, on modeling of the MID task is an example of that, where people have been choosing all these different models. It turned out that all of them were wrong and were driving possible bias. And so I think that's at least one place where. Thanks, Diane, for posting that. I'll have to have a look at it. That's a place where I think statisticians can be really helpful.
Great, thank you. And then Zhengwu, do you want to go first?
Oh, sure. Yes. Very nice talk.
Thanks.
Actually, so I'm just curious, you have presented from the data pre-processing point of view. I think, data collection, the different parameters in data collection can generate even larger impact to the heterogeneity of the data set. Have you thought about like how we can direct people who are collecting data so that they can collect data that best suited for the pre-processing pipelines we have, so that, like this whole pipeline get closed?
Yeah. I think that that's challenging because there are also trade-offs there, right? So just for example, like, sometimes you may want very, relatively high temporal resolution in your functional time series. Let's say you want to be able to do some kind of like physiological modeling and you want to have like, 500 millisecond repetition times. Those are going to come, you can get there, but it comes at a cost of both, the signal to noise ratio and the artifacts that can occur. And those costs are not spatially homogeneous, right? We know that like deeper parts of the brain are going to have a higher cost than shallower, than sort of, outer parts of the brain, for example. So it's, in part, I don't think that there's like one, you can't say, and it's going to differ between scanners as well, right? In general, I mean, there are some, as is usually the case, there are some things that kind of everybody agrees on being a bad thing to do, right? There are kind of like, guidelines that one could give to say, don't do these things. I think most of those are not things that people do anymore, in part because most, like, when somebody goes to an imaging center to collect data, they're generally going and, their imaging center gives them kind of a standard protocol. And those are generally kind of pretty well ironed out and often based on protocols from, some of these large studies that have been ongoing. Again, there are trade-offs, particularly around kind of, temporal versus spatial resolution and temporal resolution versus the artifacts that can come with it, with some of the techniques you have to do like multiband to get to very high temporal resolution. But I agree that it's definitely an important thing to be thinking about on the front end, and then to make sure that the pre-processing is well aligned with the whatever was done in the data, because there are also some like for example, head motion modeling gets different. What you have to do becomes different with very high temporal resolution because you start getting physiological things in kind of look like motion and things like that.
Thank you. Yeah, thank you very much.
Like a Lynch.
Yeah, thank you. I should preface my question by saying that I am coming from the non-clinical tomography side. We are, as a field, trying to do what you have done, but we're about 10 years behind you. So I was really interested in how kind of you've handled data management and long-term accessibility and kind of from the Pharos perspective. I was curious as to how kind of as a field, you guys have approached the idea of like persistent identifiers. That's been a big conversation among CT, aside from just like the data set having its own identifier. How have you handled like facilities and, individuals and things like that?
Yeah, we haven't really focused on persistent identifiers for things outside of data sets and people for the most part. I mean, and software, I guess we use these RRIDs for kind of software identifiers, but we've primarily focused on, like on the Open Neuro platform, every data set gets a versioned DOI. So if so basically, when you want to release data on Open Neuro, you have to create a snapshot that becomes immutable, can't be changed. That receives a DOI. If you want to add data or change data, you have to create a new version, and then that gets a new DOI. And so that's how we deal with data sets. We don't have like granular stuff within, we don't have DOIs for like individual images within a data set, in part just because it just becomes like DOI overload. And then we use ORCIDs as, and we now actually require people to log in using their ORCID ID to Open Neuro. We used to let people log in with Google as well, but for various reasons, we're moving and to basically use ORCIDs as the identifier, so we can do a better. Used to people just type their name in and they would type it differently at different times, and there's 5 John Smiths. Now we have ORCIDs to really be able to map more clearly between people and the data sets that they own.
Any other questions? So can I ask another one? So, in the field of genomics, so people will share those summary statistics, and then people can use it. And then even though we cannot run the experiments, I'm just wondering if it is also feasible for the field of neuroimaging.
Yeah. In fact, there's a whole project called the Enigma project led by Paul Thompson at USC that's basically done this. So what they do, they're doing like large scale like imaging genomics analyses, and so they don't, they can't, or they have not wanted to try to share raw imaging and genomics data between sites. And so what they do is have everybody at each site runs the same workflow, and then they share the derivative data with the kind of central group, and then that's where the genomics analysis gets done at the full scale. Yeah, so that's certainly a model that I think has been very successful in our field as well. It's not the model that we have focused on, in part just because of our kind of desire to have like full raw data in our hands when we work with it. But it's been a really successful project.
Okay, great. Thank you so much. If there's no more questions, so then maybe let me share my screen just to give you a heads up of our next seminar. So our next seminar will be on November the 5th from 12 p.m. to 1. So it's given by Professor Brian Keffel from Johns Hopkins. He will talk about brain organoid data. And then thank you so much. And then Dr. Poldrack, thank you so much again for joining us. And then thank you all. And then hope to see you next time. Great. Thank you.